tends to be a higher diversity of transcription factors that interact with each other in eukaryotic systems. I'm also going to briefly talk about how there are additional regulatory DNA regions in eukaryotic gene regulation, so we'll talk about them briefly. I want to talk about histones and how they can play a role independent of that of transcription factors in controlling access to genes. And then I briefly want to just talk about this kind of special mechanism called RNA interference. This is actually not trying to regulate the beginning process of how much transcription takes place. RNA interference is actually little RNAs that can block messenger RNAs from being translated. So maybe there are already transcripts out there, but we want to block ribosomes from wasting amino acids and actually building a protein, so we can actually interfere with translation. Okay, so let's talk about transcriptional regulation first in eukaryotes. There are some differences between prokaryotes and eukaryotes when it comes, again, to the diversity of different types of transcription factors. I'm not going to talk about that so much here. What I'm really going to focus on is the differences in regulatory DNA regions. Here's the prokaryotic system I had from the last video. Notice that in the eukaryotic system I'm not showing a cluster of genes, because remember that eukaryotic genes tend not to be clustered in operon regions like they are in prokaryotes. So in the eukaryotic system I'm still showing a promoter region. There still is something like that where RNA polymerase wants to come in and bind in order to transcribe the gene. And there could still be transcription factor proteins in eukaryotes that want to bind near the promoter region to kind of influence how RNA polymerase can come in. What makes eukaryotes more complex is they might have additional DNA regulatory regions that are actually far away from the promoter in the gene in the actual DNA code. So what I'm really trying to show here is this is just sort of unrelated DNA code right here that sort of could be thousands of bases away. These regulatory regions here from the actual promoter, and that's next to the gene. So we have kind of different names for what these regulatory regions might be depending on what type of transcription factors bind to them. If activator proteins bind to these far away regions and therefore try to get the RNA polymerase to transcribe the gene more often, we would call these DNA enhancer regions. Versus if repressor proteins bind, this isn't talked about in your book, but this is sort of been characterized now in modern study. There are some regulatory regions that are far away that repressor type transcription factors bind to, and so we tend to call those special regions silencer regions. I'm not going to focus too much on that. If you're sort of wondering how in the world can that region be as far away as it is and have anything to do with how RNA polymerase might bind at the promoter, it's a fair question, and this is very hard to animate, so I'm just going to try and draw a terrible picture instead. The idea, and this is shown in your textbook, the idea is that there might be this whole complex of proteins that kind of temporarily bend the DNA and sort of bring these enhancer or silencer regions closer to the promoter regions. And so maybe when the transcription factors bind at, say, the enhancer region, let's say that this is an activator type transcription factor, so we would call the DNA region again an enhancer region. Then when the DNA bends and some kind of protein might be sitting here to help stabilize it in that temporarily bent shape, now the transcription factor is pretty close to the promoter region and it might be able to do whatever it does in terms of promoting RNA polymerase to come in and transcribe the gene. So there's just this whole complex of transcription factors that might bind at various regions in order to ultimately fine-tune control how often a gene is transcribed, if indeed it is at all. Great. So let's also talk about a completely independent system of those transcription factors. There's really a whole other system that eukaryotes can use to control transcription rate, and that's by using histones, histone proteins, and their ability to pack and unpack regions of DNA. And so all I want to emphasize, I think I might have said this in the other video that I made, but basically chemical tags can be added to these histone proteins. I'm not going to emphasize them in my course, but two groups of molecules that are of interest to us currently are methyl groups, and they don't mention it here, but acetyl groups as well. Again, I'm really not worried about this for my particular course, but by adding different chemicals to histones, sometimes you can get those histones to change their shape and their activity. And what those chemical groups often do is they cause the histones to pack up that region that they're surrounding more, or vice versa, unpack those regions more. And why is that really important? Because that's going to affect RNA polymerase's ability to come in and transcribe at all. This little region right here in this picture is highly packed up, and so it says kind of in this caption, DNA inaccessible, so the gene is sort of inactive. You can kind of think of it as that the gene is so tightly packed in there that maybe an RNA polymerase protein wouldn't even really be able to fit. It can't even get to the promoter, so it doesn't, and it doesn't transcribe the gene. If we want to make a gene more accessible, if we want to make transcription rate higher, then maybe a cell will modify histones in another region to unpack that region more. The more unpacked it is, the more accessible the gene is. So you can kind of think of this as like, clothes in your closet, hanging up in your hanger, much more accessible. These would be like clothes packed away in your suitcase, much less accessible until you unpack them again. Okay, and a final mechanism I just want to talk about briefly in Chapter 19 is this idea of RNA interference. So we're increasingly discovering that there are far more than just 3 types of RNAs. There's more than just messenger RNA, transfer RNA, and ribosomal RNA. That's of interest to us. We're discovering really a whole panoply of different RNAs. I'm just briefly going to talk about the picture here calls it siRNA, but I'm actually going to use the term that's in your book, microRNAs. It's a little bit different from siRNAs, but not in a way that's important for me in this introductory course. So microRNAs are just kind of interest. They're actually a fairly, as they're transcribed and first built, they're actually temporarily a double-stranded RNA, which is pretty weird. All the other RNAs we've seen before are single-stranded. It's going to get cut up by a protein to sort of make it active, because in its active form it actually wants to be single-stranded. And it's going to be a fairly small series of bases, but basically it's going to pair up with perhaps a particular messenger RNA, and it's going to be complementary to that mRNA in a certain region. And when that microRNA binds to the mRNA, it's basically going to do something that prevents the mRNA's translation to construct a protein. It's almost like a cell's way of saying we really don't want that protein to be constructed right now. The microRNA might do that in a variety of ways. In some cases it might just sort of block the ribosomal RNA subunits from attaching. In other cases it might actually recruit other proteins that come quickly chop the mRNA up into little monomers again, so that it's not translated. So just another way to regulate the overall expression of genes, or sort of especially how much protein is made of a certain type in cells. What is the point of all of this? Again, the idea of conserving resources and energy, only constructing the RNAs and proteins that are necessary for a cell at any point in time. That's also going to enable cells to have more flexibility in what they produce at any given time. So maybe they can sort of express the genes and make the proteins needed for a particular environmental situation. We'll talk about examples in class. And finally, where we're really headed is developmental biology. We want to see the connection between how regulating gene expression perhaps explains a lot about how organisms develop, especially we're going to talk about multicellular human development.